Well, good morning again. I have an exciting visitor with us that we all know and love, Dr. Hande Ozdemler. And Doctor, thank you for all that you do for this community, the research that you're involved in. As you guys remember, last year, Dr. O, I'll call you Dr.
Sure.
O. Dr. O brought us a wonderful piece of artistry, a work that she did that we didn't know she had time or energy to do after she does all the research work that she does. But she brought that, it was auctioned off, and that afternoon we had anonymous donors say he would donate up to $75,000 if we could match it while at the meeting. We got to the Saturday evening process, we had auctioned everything off, and Dr. O turned to Jim Sheehan and said, "Jim, how much do we like?" And Jim said, "We need $12,000 more." And Dr. O said, "Let's do that right now, and I'll do the first 1,000." And in the next few minutes, we raised the balance of that. So Dr. O's not only in this to help us get research done that makes a difference, but helping us figure out how to raise dollars in that direction. So Dr. O, thank you for all that you do.
Thank you
all
so
that
much.
You've done, and for being here with us this weekend. Dr. I guess you're involved in so many pieces of this research, and we have followed your work with the compound NU9. So let me just ask you a very wide open question. If you were an SPF or PLS individual or family at home listening now today or listening next week on YouTube, in the research field and as we live with what we live for, what's the most encouraging news and thought you can give us today based on what you're doing?
Of course. Thank you so much for the invitation. And I think for the people who stay at home and who cannot attend here, we are still part of the family. And this is like a family get-together. And I really feel that this foundation created a family for HSP and PLS patients, and not only the love of the family, but also hope for the future. And it's not just a filled word like hope, but it's really, truly meaningful hope. And I think that I have seen you generate that over the years. So I'm humbled to be part of this family, so thank you so much for including me. And I see research exponentially growing. First, for example, we did not really know about the genes, the mechanisms. Now we know more than 80 mutations, 150 total mutations. The genetics of the disease is better understood. And with that, the mechanism, the underlying mechanism of the disease is much better, precisely understood. So we begin to understand why each patient developed the disease. What is the underlying cause? Once we know that knowledge, then we can actually formulate effective long-term treatment strategies. And those are emerging with gene therapy approaches, ASOs, and with the gene therapy, you have seen that there's a clinical trial already initiated with children. And that's an amazing achievement. And the ASO is an important topic, and you know the ALS field, SMA fields, they are making important strides in that. And I think HSP and PLS is prime targets for those because we know the mutation. And I think that is an open field that's coming up very fast, very quick. And I think that's why it's important to be involved with the foundation as early as possible and as much as possible, because when there is change, there is momentum, you need as many people in the game with you. So I think that's very important. In addition, for the first time now, we identified a small molecule, a compound, NU9, as you said, that improves the health of diseased upper motor neurons. I mean, that is game-changing. We did not have any compound that improves the health of upper motor neurons. Now we have it. And most importantly, now there's a company, Akava Therapeutics, which you can check on its website, says our primary target diseases are hereditary spastic paraplegia and primary lateral sclerosis. We did not have a company out there fighting for those patients before. Now there's a company, there's a small drug, and that NU9 actually got approval from FDA to start phase 1. I mean, can you imagine how exciting that is, that we are going into clinical trials, and that within a short period of time this drug will be tested on patients, on healthy volunteers first on phase 1. And these are very exciting times. And plus, thanks to your foundation, Spastic Paraplegia Foundation, the clinical centers are forming a cohesive unit, and then they are getting ready for the clinical trials. And that is extremely important because this is a rare disease, and some patient is seen in this hospital, the other patient is seen in another hospital, but there was not really too much teamwork or working together between those centers, and now you establish that. There are at least 10 different centers working together. They're getting ready for the clinical trial. They're looking for the, they already initiated the clinical trial. So they're building a platform for the next clinical trials to happen. And I hope NU9 would be the first one or the second one coming up in the pipeline. But once you form the pipeline, you will have many solutions going through that pipeline. Forming that pipeline was the tough work, and that's what you achieved, and I thank you so much.
I appreciate everything you just said. Those of us who work with the foundation and work with foundation members and potential contributors, we get asked often, well, explain to us what has our research accomplished? What has the money we've put into our research accomplished? And those of us who are not scientists and not researchers have a very difficult time specifically answering that question. But what I think I just heard you say is all the research that we and others have paid for in the past have gotten us collectively to the point where these exciting things may be just around the corner as we talk about compounds like NU9, as we talk about additional research in those areas, as we talk about clinicians who are assisting in creating that platform for clinical trials. So all the elements seem to slowly be coming together. And I'm sure none of us like to hear what I'm about to say, but it's just the way it is. The way this happens at the next level and the next level, it's a matter of money. Whether that money comes from grant resources, that this foundation is working diligently to try to access better roads to NIH and other sources that have substantial amounts of money. But in the meantime, the contributions that all of us as members of this foundation and others make continue to be important as we build those bridges between here and NIH and clinical trials. So it's tough to say we need to continue to raise more money, but we got to get across these bridges to get to the point you're talking about, correct?
That's correct. That's very correct. So I know, with the foundation, the research grants that you have given to me, to other labs, are exceptionally important. And I know for the donors and patients out there, they may think like, where is this money going and how much contribute is this making? But I am happy to tell you that this actually makes an immense contribution, because the knowledge that we generate with those foundation grants lays the foundation for us to go for NIH grants, for bigger grants. If we don't have that support at the very beginning, we have ideas, but then the ideas need to turn into preliminary results. And if those preliminary results do not come about, then you cannot go and ask for NIH grants, for bigger grants, and so forth. So that's why the foundations are extremely important for those great ideas to nourish. And, for the patients out there, for the friends of patients out there, I think every support that you show to the foundation helps the scientists and the doctors to eradicate this disease, because we have to put money towards knowledge. And the more knowledge we have, the less disease we have. And that's why I'm here to help as much as I can with my artwork, with my donation, with my time, because as I said in the beginning, we are family. And when family gets together, family solves the problems. And I think this is the time that we solve the problem and we see a better future for everyone.
And this family is larger than even SPF. I mean, for instance,
I think so.
It doesn't matter to me which foundation or which foundations fund and find a cure. Those of us who have these diseases just want a better tomorrow.
That's true.
And so SPF came into existence, and its primary purpose today is to help fund this research. So we appreciate any contribution folks can make to go toward that purpose. But there are contributions in other places, and if those contributions go toward your research to find these cures and these treatments, we're thankful for that as well. So we invite you to consider anything you can do, whether it's with SPF or with another foundation that directly supports this kind of research and work toward treatments and cures. Dr. O, we love you. We appreciate you. We thank you for being here, and we look forward to continuing to work down this process. One of these days, we're going to come to an annual convention, and we're going to have balloons and champagne like a New Year's Eve party, because we are going to have found one treatment that makes a difference in people's lives, and it's going to be because of folks who contribute and people like you who are doing this research. So thank you for being part of our team and part of our family. We love you and we thank you.
Thank you so much. Thank you.
Thanks for being with us this morning.
Thank you.
Guys, you heard a great explanation. Dr. O said it very well. She's on the front line of that research, and so thank you for listening to that. And Dr. O, I'm going to invite here in just a moment Dr. Fakari to join us, and we're
Sure.
going to get him to talk to us about that clinical platform that you
Sure.
were talking about just a moment ago. With that, I want to say thank you to our PLS folks who are here. I'm wearing, as you all know, I'm an SPF and I have HSP. But we have the largest group of PLS folks that we've ever had,
Sure.
and they came armed with PLS T-shirts. Norma and I were honored that they shared those with us, and so today we are supporting this PLS group that's here, and those of you that are home. And we want you to know that we're going to do all the research we can, both in the HSP and the PLS front, to find some answers.
Thank you.
So Dr. O, thank you, and I'm going to call in Dr. Fakari. We're not even going to leave the set here, just let him come in and take the microphone. A very good transition, as Dr. O has talked about this process that moves in the direction of our SPF CERN and the common work that you guys are doing, Doctor.
Sure.
We had, I shared with our group a little bit earlier this morning that we had a little bit of technical difficulty at the end of yesterday. I had just told them that we were going to turn to early child onset, and we didn't get to do that, so I appreciate your coming to visit
absolutely.
today.
Very happy to. Thank you for having me.
I would just ask you to share with our listeners at home, and of course those who may listen next week on YouTube, if you can just summarize the work and the discussions that you and the parents of the children were having yesterday. We would love to follow up on that.
Sure, absolutely. Very happy to. And I'll start out by saying that I think it's wonderful that the SPF is supporting this effort. I think there are a couple of topics, and this is what the discussion focused on yesterday, that are very, very important to parents with children with hereditary spastic paraplegia. So we talked about the link to cerebral palsy, the delay in diagnosis for many, many children. We talked about our approach to symptomatic therapy, and then we spend a lot of time talking about sort of community support, how parents support each other, and we talked a little bit about research. So those were the sort of the big areas we talked about.
Last year we celebrated as we created the CERN, and so share with us, if you would, just kind of a quick summary of how things have gone in year one and where you see that going in year two.
Yeah, very happy to. So a high-level summary, the Spastic Paraplegia Centers of Excellence Research Network, or CERN, really was created with the idea of building a national network that can support clinical trial readiness for hereditary spastic paraplegia and PLS. And year one very much focused on doing the foundational work for this. So I always think about these larger projects, if you want to use a metaphor, like building a house. You first build a foundation, and that's what we did in year one. So we got together, we organized a group of 10 centers around the country, 3 centers who focus exclusively on childhood onset HSP, 7 centers who see patients in the adult age group. And year one was really spent on organizing the network, developing a central research protocol, which is a key step, getting this approved and rolling this out. So we're very excited that just a few weeks ago we were able to enroll the first patient at the Boston site. We're up to 5 patients as of today. So slow start, but we're starting. And we hope that the other sites will come online very soon.
Well, we're very excited about that process, and for our listeners at home and in the future, as the CERN group meets monthly to talk about progress and coordinate activities, 3 of your board members, Norma and I, and John Cobb, our research chair, and Frank Davis, a past president, we've been invited to sit in and participate, and it's good to just listen, and every now and then we can provide a piece of information that
Absolutely.
is helpful. But it's good for us, Doctor, to know the foundation that you're laying, the plans that you're making, and how that's consistent across all 10 of those clinics.
Yeah.
And as we hope that foundation builds more clinics in the future,
Absolutely.
so that we create nationwide this opportunity for, we hope, a quicker diagnosis
Yes.
and quicker access to clinical trials as funds become available and that process moves in that direction.
Absolutely, and I think if I can add to that, and also building, I think, expertise in clinical care. I think you're absolutely right. This is just the first round. We have 10 centers. Hopefully in the second round there will be many more, so we can really, represent most parts of the country. And I think over time what I've seen is that these kind of initiatives, they lead to more research, more awareness, more funding. They lead to better clinical care because people specialize in the care for individuals with hereditary spastic paraplegia. So I think all of these benefits will take time to build, but they will become, I think, available to the community very soon.
As Dr. O and I just were commenting a moment ago, those of us who are on this board, and we're not scientists or doctors, and we get asked often, how can you tell us that our research that we have funded in the past has made a difference in terms of where we are? And I would just ask you maybe for your response in that regard.
Sure, it's a great question. I think funding through the SPF has been instrumental, not just over the last year, but over many years, in moving the field forward. And it's really difficult to give this a metric, right? But I think the things that I'm excited about are clinical trials coming along? Are we seeing the basic science that will lead to clinical trials eventually? Are we seeing earlier diagnoses? And I think all of this has been supported through research supported by the SPF. So in science we use metrics. We use metrics like publications, metrics like patients enrolled in studies and things like that. And all of that has been trending in the right direction with the help of the SPF. So I think those are sort of the things I would point out. And then, there are a couple of milestones achieved already, I think. For us in Boston, it was a major milestone to have a natural history study for all types of hereditary spastic paraplegia, something that didn't exist a few years ago. We are able to provide patients and families with unknown types of hereditary spastic paraplegia with the latest in genetics. And all of these things are only possible because there's a strong community and funding provided to these research studies.
And in terms of past funding and future funding, as we were speaking a minute ago, this family is a big family, and there are a number of foundations and places where folks can contribute toward HSP and PLS research. SPF is certainly one that's been around for a long time, and we solicit and we ask for every contribution we can and ensure that that's going to go toward this research process. But we also put our arms around and cheer on other foundations because in the end, doctor, if you find a treatment that makes my life better, I don't care where it came from. We just want to see people's lives made better, and we appreciate the work that the new CERN group is making in building that block, and the research that we look forward to with clinical trials, hopefully not far down the road. I'll ask you one last question, and then I'll let you go. But I asked Dr. O this question as we started.
Yeah.
What's the most optimistic, enthusiastic encouragement that you might give people in terms of where we are and where we go next?
Yep, that's a great question. There's a lot of things to be excited about, I think. But when I meet with families in clinic every week, a lot of what we talk about is the fact that we're at a day and age where we have novel genetic therapies available, therapies that not only treat the symptoms, but really get to the root cause of the problem. And this is something that didn't exist when I first started going to medical school. So science is advancing fast. Some of these advances haven't made it to HSP yet, but will make it eventually. And that's what I'm very excited about.
Well, thank you for your work. Thank you for your partnership here with us.
Thank you.
And we look forward to continuing work together until we cross a finish line that'll make a difference.
I very much appreciate it. Thank you
very much, Doctor.
Thanks a lot. Thank you.
Well, folks, we are about ready to turn back toward the podium. We thank Dr. Fakhari and Dr. O for joining us. It's great to have their expert input on what they're doing and where we're going in the future. But at this moment, we're going to turn it back to Tim up front and proceed with the next part of this program. Thank you for joining us. We'll see you later.
A second, get everybody back into their seats so we can get ready for our next presentation. Hey, real quick, I wanted to point out, we have these bracelets at the tables for you guys, and you got some in your bags and stuff. These are really great tools to hand out to people when you meet somebody new, and somebody asks about what your condition is or how you're feeling or what's going on. You tell them, here, peel off a bracelet, hand it to them. It's got information on it. You can tell them to go to the website to take a look and learn about HSP and PLS and the Spastic Paraplegia Foundation. This is a great little tool to spread the word about us and what we do and about you and how you're doing. So we're moving right along. Coming up next is Dr. Pia Marini. Here she is.
Hello, everybody. I'm Emanuela, and I'm very happy to be here this year as well. It's my second time attending the SPF meeting. So I want to first thank the organizers for inviting me and for putting together an amazing meeting. I've really enjoyed it so far. So today I'm going to talk about some of the data we gather on the gene therapy that we are working on for SPG4, as well as some new ideas on how to also promote functional recovery on people that are already affected. In the lab, we mainly study SPG4. Dr. Bass talked extensively about it, so I'm not going to repeat too much of what you already learned from him earlier today. I just want to point out a couple of things again, that SPG4 is one of the most abundant form of the autosomal dominant HSPs. It accounts for 40 to 60% of all the HSP known forms, and there are more than 200 mutations in the SPAS gene that cause SPG4. And the SPAS gene encodes for this protein you've heard from him that is called spastin. And what I want to point it out again here is this interesting thing that spastin encodes for two isoforms called M1 and M87, that they differ a little for their location in the cells as well as for their function. In the lab, we made a while ago, before I even joined the lab, this mouse model that we call the SPAS-C448Y. And this model was made by Dr. Bass in order to study the gain-of-function toxicity. So we introduced the human-mutated spastin in addition of the endogenous spastis on the purpose to test the gain-of-function hypothesis. So Dr. Bass already talked about it, and again, I'm not going to repeat myself. I just want to introduce the model that then we used for the gene therapy, as this model show behavioral defects and anatomical changes that are similar to the one that are described for the SPG4 patients. So that's why we decided to move with the gene therapy with this model in particular. So let's look a moment at the work we have done on a preclinical model for SPG4 using this animal. First, I want to point it out that the gene therapy is a concept that started earlier in the 70s, and throughout these last few decades, there were numerous attempts and numerous studies done on viral vector-based gene therapy approaches on a number of different diseases. All these studies has demonstrated us how effective the gene therapy is and clinically safe. So when we started to think to the SPG4, we thought that using a gene therapy approach to replace the mutated bad gene with the correct one was a therapeutical avenue that can be pursued. So we collaborate with the team at UMass. They are experts in gene therapy, so they help us in designing and in strategizing how to use this vector, we have a dual vector in which there is a component that can recognize the endogenous spastin and so remove it, silence it. And then the other component of this vector, it helps in re-expressing the correct spastin. So it's a classic gene therapy approach in silence and replace the bad gene with the correct one. How did we do the testing? So we started with pre-symptomatic animal at P0, P1, so they are very small pups. We injected this animal with our spastin vector. You can see here they were going under anesthesia. And then we let them grow, being in their home cage, and then at the 3 months and 6 months of age, we tested them on the catwalk because these are two of the timeline in which we start to see early symptomatic signs of gait alterations and later symptomatic. The catwalk is this, is this device that has been used for a number of studies on rodent, especially to assess gait alteration, and is an automatic device, so it's very unbiased, and you can use to draw a lot of different parameters. After the 6 months, we collected the tissue and we move on with further investigation. So first of all, we look at the spastin expression. I'm not going to stay too much on this slide because I just want to show you how our very first attempts in understanding the level of spastin in the disease animal as well as the animal that were injected with the vector were, and how difficult and hard was for us to detect the famous M1 isoform. So thankfully, we have recently received this first batch of polyclonal M1 antibodies, and we were very excited to test on this animal because it's just so important for us to understand how much of this spastin we are replacing with the vector. So as you can see here, there is a much better Western blot showing you the M1 band, bright and crisp, and then we have the M87 band also present, and GAPDH is used to control the amount of protein we are introducing. Now, the interesting things here is, as you can see in the B and C graph, the M1 and M87 expression is enhanced in SPG4 animal, specifically, or actually particularly, the M1 expression is really high and significantly higher than the M87. But when we injected the animal pre-symptomatically with the spastin vector, what you can see here with the yellow bar graphs is a restoration of the spastin level to a normal condition, suggesting us that with this vector we are able to restore the level of spastin just as much as needed, so not too high or not too low. And it has been one of the most exciting results we have had so far, and it's also highlighting how important it is to have the correct and the right tools in order to study this disease. So axonal degeneration can be studied using immunohistochemical technique, and HSP is known to have cortical spinal degeneration as one of the main characteristics. So how can we assess axonal degeneration? We can use markers that helps you in visualize what's going on in the tissues. And one of these markers are neurofilaments, which are architectural structure. And it has been shown in a numerous of studies on other neurodegenerative diseases that an increase in these phosphoform of the neurofilaments is associated with degeneration of the neurons. So we decided to use these as a marker to assess if also in our animal we have degeneration of the neurons, as well as if what happened when we introduce the spastin vector on this animal. As you know, the main feature is degeneration of the cortical spinal tract. So in the mice, how can we look at the cortical spinal tract specifically? Here is a cartoon of the cross-section of the spinal cord of an animal. And as you can see in the red circle, there is where we focus our analysis, because in the dorsal column of the spinal cord of this animal is where the cortical spinal tract pass by. It's a little bit different than human people. So how do you visualize this? As you see, you have these green dots that are stained for the phosphoneurofilament, and in the SPG4 animal, these green dots that represents the axon are much brighter. So that means this phosphomarker is increased, and that is telling us that these axons are degenerating. And you can see here in the graph. But when we use our spastin vector, these bright spots are not anymore as bright, and are actually more closer to what is the control condition and in the healthy animal. Okay. Then we assess using behavioral device, as I said earlier, if like we look at the behavior to understand the gait alteration that are in this animal. And one way to look at them is using the catwalk that I mentioned earlier, because the catwalk has all these different parameters that can help us in understanding how these animals are walking and if they have problem, right? So I'm not going to go into the specific of each of these parameters. I just want to point, we choose those four mainly because if you want to describe a way to how the animal work, or also how people work, is by looking at these parameters. And these parameters were actually also used to assess gait alteration in HSP patients. So one more reason for us to use this parameter and look specifically at this parameter when we use the catwalk to assess the behavioral phenotype of this animal. Again, all these graphs, I just want to point out a few things and not going too much into the all little details. But as you can see, the wild-type animal, they don't show much of a difference between their 3 months time point and the 6-month time points. They just walk fine, they are okay. When you look at the SPG4 animal, you start to see an increase in this bar graph that is telling you these animals are having problem in their walking condition, and this is represented by almost all these parameters. These issues in the walking condition are like present at 3 months of age as early symptomatic stage, and they get worse at the 6 months time point. But happily, we saw that if we introduce the gene therapy vector on this animal as early as P0, P1, so when they are just born and they don't show any symptoms, we are able, as you can see by all these graphs, bar graphs appearing, that these animals, they don't show anymore these phenotypes. So that is telling us once more that this vector is helping in preventing the manifestation of the symptoms. So this is all very exciting, and we are still working quite hard in refine any other tool we need to look at to make sure of like how this vector is working and how can help, and like we still have some check mark to do to make sure to present a comprehensive work. But it's all very exciting in seeing that as early as P0, we are able to prevent the manifestation of some of the main symptoms that are recurring in the SPG4 patient. Last year, when I was here and I was talking about some of these data, I got a question that since then, I guess, it stuck a little bit in my head. And one of the questions was like, and this question was essentially, well, that's cool, interesting, but what this can do for the people that are already affecting, or how this can be used on people that have already symptoms. So we talk a lot in the lab in how we can address this for the community. And so that's why the last part of this talk is just few ideas that we got in the lab that we want to test on SPG4 to understand if we can promote functional recovery on symptomatic SPG4 patients. So at Drexel, we are very lucky because Drexel is also the home of the Marion Murray Spinal Cord Research Center, and we have some of the best researchers in the spinal cord injury there, so that help us in discussing in a more broader way our own project and actually gain knowledges from them and try to apply what they learn in the spinal cord injury field to HSP potentially. So, in the last decades, the exercise therapy is one of the one has shown the most promising data to promote functional recovery after spinal cord injury. The exercise therapy induced the production also of neurotrophins that in the end promotes the sprouting of axonal branches, so you can restore the route to the tissue target. Moreover, here it is. Treadmill exercise has been long used as a therapy for spinal cord injury rehabilitation. It's a very easy essay and is like, and it has shown a lot of promising data in promotes and restore functional recovery in spinal cord injury as well as in other disorders, not disorders, as well as in other, like in traumatic brain injury or stroke. And it also showed that can promote cognitive recovery and reverse a sign of inflammation that in general some of the signs you see when you have degenerating neurons. So what we thought is that maybe we can combine the knowledge that we get from the spinal cord injury field and apply to the HSP by using as Dr. Bass said earlier, a combinatorial approach in which we can use the gene therapy on symptomatic animal, in this case, to stop the progression of the disease, because I envision that by just injecting the animals, in this case, at symptomatic age, it might not be possible to have a full recovery, but it is possible to stop the progression at that situation. But maybe what we can do, as I just said, is combine the gene therapy approach with something like the exercise therapy in order to have maybe a little bit more of just stopping the progression of the disease, and maybe we are able to restore some of this function. So here I just highlighted like a brief bullet points, although they are not bullet points, it's just to show you how we are thinking to address to that. So what we want to do for the next step while completing all the work on the pre-symptomatic animal is starting to inject the symptomatic animal and then have another cohort of animals that are also receiving exercise therapy. So we are going to look at the early symptomatic animals as well as late symptomatic animals. As you saw before, at 3 and 6 months is enough for us to address these two stages. And we are going to inject them with intracerebroventricular injection with the vector. What we will do next, we will use the treadmill exercise by using another device that is called DigiGate. And the DigiGate is similar to what I showed you before because it can help us in assessing gait parameters, but it also can be used as a treadmill so we can exercise the animal on these while getting also data, meaningful data on how their walking condition are over time while exercising. And then we are going to look at the morphology of the motor cortex on these animals. So we are going to collect CNS tissue and look at like the morphology in order to study using immunohistological technique again, plasticity and axonal sprouting, as well as we can apply tracers that allow us to visualize a specific tract and look at if there is any sprouting happening after exercise therapy, as well as we are going to investigate the expression of neurotrophic factors that are beneficial in the restoration of these functional recovery. And in particular, we are looking at some of these neurotrophic factors that have been shown in literature that promotes restoration and axonal sprouting, but like we will look even more deeper in, for example, also molecules that are called morphogens, so that they gives to the neuron some cues to understand, basically, they guide the neurons that are regrowing towards their target. So all of this is just our preliminary idea and attempt to try to understand how to help also the community that is already symptomatic, and it's just like a further step towards understanding how to give tools to the community and like get some beneficial effect. So with that, I would like to thank all the people that work with me on the gene therapy project, our collaborators at UMass, especially that they help us in designing the vector, Dr. Miguel Esteves and also Dr. Heather Gray-Edwards. Obviously, none of this would be possible without the funding agencies, the Cures PG4, the SPF, and the fellowship that I got from Drexel University. And thank you all for your attention, and I'm happy to take any questions.
Pardon me if my question is elementary, but my, I guess, version of hereditary spastic paraplegia is I have SPG4 and I have like a deletion in my gene. Mm-hmm. But I hear a lot of you guys talking about like mutations. Is that different?
Yes and no, in the sense a deletion is anyway considered a mutation. It's different. So like a deletion means that you have eventually a stop codon that is like not, let me see if I can find simple. So basically, you are not producing a full-length spastin, but just a piece of it.
Thank you.
No problem.
Any other questions? Okay, we're going to take a quick couple of second break here, and we're going to make a changeover for our next presenter. Dr. Primarini, thank you very much.
Thank you, everyone.
while I got a couple of seconds to fill, which you know I can do, remind everybody, remember, we still got the potato pants out there, so if you haven't had a chance to put those on and walk around in those, please do so, okay? Because it's a really, really cool thing to do to kind of feel and understand what it's like to have an upper motor neuron disease that makes it difficult for your ability to walk. Also, remember, there's still time for raffle tickets. Yeah, I'm telling you now, later when you see what we're raffling off, you're gonna wish you had more. I'm just saying, to increase those chances. Remember, you can't win if you don't play. We still got t-shirts out there for sale. We got some retro-looking ones, which are really cool. We still got some of the Tampa ones for this year, so make sure you get out there and get those, okay? Also, we have the caregiver items that we talked about earlier out there at the table you can take a look at, that things that can help people throughout their day. We brought those so you can browse those and find those out there on Amazon and stuff. So we're getting lined up here. We're getting ready to go. We're looking pretty good. Here we go. Are you guys ready? Here we go. Ladies and gentlemen, Dr. Chung. Here he is.
Good morning, everyone. It's an honor to be invited to the SPF conference again and to present a small portion of my work in the lab. I study SPG4 in the lab, and it's just a small portfolio of my research projects. I wish I had more time to share more data with you guys, but unfortunately I have only 20 minutes to go through some really important things and updates I want to deliver to you guys. And would like to take more questions from you guys, and that could be very, very informative and inspirational for my continuous research on SPG4 and HSP. So I'm going to shift the gear a little bit today from animal models to human stem cell models. So my lab is dedicated to use human stem cells, particularly patient-derived stem cells, as models as well as a therapeutic sort of approaches to study the mechanism of the neurodegenerative diseases, as well as sometimes to treat neurodegenerative diseases. So what are the stem cells? I guess everybody here might have heard stem cells before. Stem cells has really two major characteristics. One is the stem cells can be reproducing itself. We call it self-renewal. So if you have stem cells, the stem cells can actually reproduce identically to daughter cells that represent exactly the same thing as a mother cell. The other really important characteristic about stem cells is it can differentiate it into functional and mature cells that carry out major functions in your body. For example, a stem cell can become a lung cell, can become a brain cell, can become a kidney cell. So these are the stem cells, the characteristics, and they're so important. And then kind of our body was built up from different stem cells when we were just a zygote, starting with a zygote, with the sperm and oocyte and combined together. The old dogma in the stem cell field is when your stem cells differentiate into the somatic cells, we call it functional and mature cells, there's no way they're coming back to the stemness. But the new trick is, for the past 20 years, we learned a lot from transcription factors in our nucleus that can reprogram our somatic cells. For example, our skin fibroblasts or the blood cells you can draw from your peripheral blood, and then using some strategy to turn these cells into a stem cell stage. So it's a reverse your natural differentiation process to go back. And that really kind of is revolutionary in the biological field. Now we can turn, let's say, my skin cells into my embryonic stem cell stage, and then I can develop these cells into any type of the cell, compose my organs or tissues in the body. So technically, we call these stem cells induced pluripotent stem cells, and that's what I'm going to use this time as iPSCs. So why do we need human stem cell or HIPSC-based model for HSP or PLS? That's a good question. And for years, and you hear today and yesterday a lot of talks from scientists using transgenic rodent models. They're really powerful and very, very useful to study disease mechanism, to screen drugs, to kind of carry out a lot of experiments to answer these questions for us. However, there are inevitable hurdles and limitations of the animal models, especially the commonly used transgenic rodent models. The first one is create each animal model. It's very costly. It takes like $20,000 to $30,000 for one strain of the animals. And that's just one mutation, let's say, if you want to put it in. The second thing is there is a big difference or nuance, I would say, wouldn't say big, a nuance difference between the genetics from the animals, especially rodents with the humans. And also there are physiological differences as well. When I say genetically, it means the proteins that compose our body, let's say spastin, elastin, or other SPG4 or SPG mutations, the protein you have, there are nuance difference in their sequence. And these nuance difference can be actually significant in the end to carry out the functions of the proteins. So the proteins in the rodents doesn't really equals the proteins in the human body. And also, particularly in the human cortical development, especially the motor cortex, it's way more complex than the rodent models. And last but not the least, the CST, the cortical spinal tract in rats and mice are very different than the humans. They innervated differently. They don't, in humans, most of the cortical spinal tract, they innervate the lower motor neurons directly, but that's not the case in the rodents. And the third thing is there are a lot of clinical trials that failed on the drugs that showed really good efficacy in animal models. We still don't know why, but that's like the fact we have to face with all the drug development. The last but not the least, I think is a really important point here is you can't use animal models to study undiagnosed or sporadic disease. That means these disease cases are without known gene mutations. If you don't know the mutations, how do you model that in the animal model? You can't. But the human stem cells make it possible, especially from the patients, because you don't really need to know what the mutation you have. You just need the human cells from the patient. So more rationales to use the human stem cell-based models. Of course, I covered 50%, more than 50% HSP and PLS have unknown genetic mutations. That makes a perfect sense to use human-derived stem cell models to study the disease. The other thing is we have a really, really low amount of the post-mortem patient brain and spinal tissue to kind of study the disease. But that can be changed by the human stem cell technology because you can literally create your patient tissue, although it's probably more embryonic or developing than the mature and adult, but it's from the patient, as the brain-like tissue. The third one will be these taking the cells from the patient, really realize the personalized medicine and precision medicine that can be reflected on drug screening and toxicity test to see whether this drug is particularly effective on this patient versus the other patient. What to do with that, to use your own cells, to use your own cell-developed brain tissue to test these things. And how are we going to planning to use the human stem cell iPS cells to study HSP and PLS? So here we go. We generate human brain organoids enriched in upper motor neurons to study these diseases, particularly in my lab to study SPG4. What are human brain organoids? Those are miniature brain tissues that are small brain-like structures with essential brain cells. For example, upper motor neurons and the supporting cells like glial cells and different type of neurons in your body as well. And then they are not just mixed together, they actually are arranged in an orderly and a complex manner, really kind of give you some presentation of how the human cortical tissue developed. So we have two strategies in my lab actually to create two sets of the stem cell banks. So one we call this isogenic stem cell bank. These isogenic lines were developed from one healthy donor. And that could come from the blood and could come from skin biopsy or the skin fibroblast. We could grow them up, and then we can use our technology to turn this into embryonic stem cell-like stage, we call iPS cells. At this level, now we can introduce different mutations by the Nobel Prize winning technology called CRISPR. So that is so powerful because from this one donor cell line, we can create as many different mutations as we can to represent all the people here, the mutations you have, using the CRISPR technology, but with the same genetic background, which is we call identical genetic background, except your point mutation or other mutations you have. And then we can turn these iPS lines into human organoid, brain organoids enriched in upper motor neurons for disease modeling. This is so powerful because now it gives us the tool to study genotype, phenotype correlation. And what is the genotype phenotype correlation? I heard a lot of people are talking, I have SPG4 mutation, but I have a deletion. I have SPG4 mutation, I have a missense. Are they the same? Are they presented in the same way as a disease mechanism? There used to be a lot of questions on studying these things, but you can't really put them into a very rigorous studies without having the same genetic background. Because as human, all of us have a different genetic background. You don't really know what causes the diseases from the mutation or something else, because we're all different. But in this technology, we can actually study the same thing, under the same genetic background, just that mutation you have. The other set we have, starting to develop in the lab, is we take patients' fibroblasts from different sources. And these are patient directly from the patient, the skin fibroblast. Now we turn into iPS stem cells. Now we make organoids. And these organoids are actually just like similarly to your brain tissues, your own brain tissues. And then these are from different patients, from different type of mutations and different gene mutations, we can do that. And this really gives us to precision medicine or personalized medicine because these are directly from you patients. So here are, if someone can hit the first figure and then let it run. So these are how we generate the organoids in the lab. We have a bioreactors. You can see each of these little white dots here. They're representing the organoids and the brain tissue, brain-like tissues we're growing in the lab. The left one is a little bit younger. You can see they're smaller. The right one is a little bit more mature. We now can grow these organoids up to 12 months in the lab. But today we're going to talk about some of the organoids we used for analysis. These are from 3 months, 6 months, and from 12 months. So after we, develop this strategy to develop the human organoid, brain organoids or cortical organoids enriched in upper motor neurons, we want to characterize it to see whether these are having the characteristics of our human brain. And here is the first picture you see here. You see these little red loops. Those are neurogenesis site, and which means that your neural stem cells developed these little loops that give rise to neurons and or nerve cells and glial cells, which are supporting cells, and to kind of build up your brain. These are the little bit older organoids. You can see the mature nerve cells migrated out of the regenerative zone, we call this, and then just form this kind of cortical plate-like structures with axon like extended, like the cortical plate structures. And we really want to make sure our organoids are actually representing upper motor neurons here because that's the disease we're targeting. So we use a CTIP2 as the nuclear markers to see, do we get enough CTIP2 positive upper motor neuron-like cells? And we do. You can see here we have, we quantified it's probably 20% to 30% of the neurons we're getting are CTIP2 positive. And you can see these, another gene is SPG1, it's called L1CAM. That's also highly expressed in our brain organoids, which represented these are very close to what we have so far. We call this cortical organoids enriched with upper motor neuron region. And I'm going to present some of the studies we did with the disease phenotypes, and I don't have time to go through all of them, so I'm going to skip the set 2 and focus on set 1, which is isogenic HIPS lines, and we're not going to do any deletion today. We're only going to just focus on a truncated mutation S245X and a missense mutation C448Y. I want to remind you guys these are isogenic ones, so they have identical gene background, genetic background, only the gene differences on spastin. So one has a truncated one has a missense one. So we analyze the 3-month-old organoids to start. You can see here are the CTIP2 stainings, which are reflecting our upper motor neurons in these organoids. You can see control have a robust production of CTIP2. However, in both truncated mutation and in CY mutation, the missense mutation, we are losing the CTIP2 cells, which are very unexpected in our previous, before we did experiment, we were not expecting to see there is a loss of the CTIP2 cells in our organoids. And the other difference between these, I call this genotype-phenotype distinctions, is we did see the difference between the truncated mutation and the missense mutation, as the missense mutation have enlarged the cell bodies, which another sign of the cells are not doing well, doing worse, not just to have a lower amount of the cells, but these cells are sort of dying. Not all of them, a portion of the CTIP2 positive cells are actually undergoing apoptosis. Which really surprised me is this figure here. The GFAP is a marker for astrocytes. I'm not sure how many people are knowing astrocytes in our brain. We have nerve cells which carry out a lot of functions, but consider astrocytes as a supporting cells. With them, you will not have functional nerve cells to do your job. So these astrocytes are actually having 2 different directions in our mutations. In the truncated mutation, we have very, very hypertrophic astrocytes, which means astrocytes are not developed well. However, in the missense mutation, the CY mutation, we have hypertrophy astrocytes, means these astrocytes are hyperactive. They're actually probably more inflammatory. So that makes probably the worst of the condition of the missense. So we are still investigating on these issues on astrocytes in SPG4, which is not really considered as sort of a big studying point for a lot of scientists here, but I would like to go deeper or dive deeper into the mechanism why the astrocytes are showing these 2 different directions in the 2 different mutations in SPG4. Last but not the least, for the genotype-phenotype distinction, we see there is a different level of degeneration happens in the 12-month-old organoids. When you see here in the middle is the truncated mutation, and then all the way to the right is the missense mutation. The truncated mutation, both of them, truncated mutation and missense mutation, shows a little bit increased markers for neurodegeneration. We call this SMI32. And these are our markers, or we consider that as a readout for our therapeutic interventions in the next slide I'm going to show you. So I hope I can deliver this message as in the truncated one and in the missense one, they both show the degeneration markers. However, the missense one again shows even worse degenerative markers than the truncated ones. So now we get into the therapies. So previously, collaborated with Dr. Boss, we find HDAC6, the histone deacetylase 6, is probably a therapeutic target for SPG4 specifically. And then I have this wonderful models, then I can test whether these HDAC6 can actually act as the therapeutic target with small molecules. So what I did is I validated this HDAC6 change in these mutated organoids. Interestingly, at 3 months old, only the missense mutation shows there is abnormal increased functions of HDAC6. However, in the 6 months old, the truncated one catching up. So you also see the abnormal increase of this HDAC6 activities. Very interestingly, we obtained human brain tissues from one male post-mortem brain and one control, age-matched control, the same region, and we run the HDAC6 activities in both. Interestingly, I want to point it out, these HSP patients we got from NIH, they actually don't have the SPG4 mutation. And we did our best to sequence this, the brain, and we find there is one very rare SPG4 mutation in this brain. It's called CTC1. So it's a newly found HSP gene, and actually it's mutated in this brain. However, no matter what, we do see an HDAC6 and increased activities in a non-SPG4 brain. HSP brain, which is quite exciting for us to point at HDAC6 might be a universal target for the whole HSP spectrum, but it's very preliminary. And we use our drug called tubistatin that's the drug that can inhibit activities of HDAC6. We want to validate whether this drug is doing what it's supposed to do, and we see, yes, it is. It inhibits HDAC6 dramatically. Now we look at the phenotypes. So we put this drug in different concentration, different durations of the preliminary data, but I'm not gonna bore you guys with all the experiment conditions I have. I just want to show you here with the one examples with the missense mutation. We put the drug on. You can see very obviously the degenerative axons marked by SMI32 was much reduced with tubistatin treatment, which is inhibit the HDAC6. So hopefully I convinced you guys the novel human models derived from patients are really essential to model the HSP and PLS and other neurodegenerative diseases. And particularly we find a way to culture these human stem cells and to upper motor neuron enrich the brain organoids, and that's unprecedented. The second thing is, I think I delivered some preliminary data to see there is a genotype-phenotype distinctions on the case of SPG4. We think missense mutations, especially what we're looking at, has a severe phenotype than the truncated mutations. The third, but not the least, is the HDAC6 we think is a very promising therapeutic target. And we actually not only see that in our SPG4 cases, but in the different HSP cases that's increased, and are really offering a promising therapy. So the ongoing project in the lab is to test new druggable targets to treat HSP and SPG4, and we have several novel targets now identified by our high throughput analysis in the lab. We're testing it actively. The second thing is we want to collect the SET2 IPS, which is from the patients, to really expand our portfolio or our reservoir of the IPS banks. The third one is we're actually collaborating with Dr. Boss and other people as a multi-PI collaboration to test the gene therapies using our human patient or patient-derived brain organoid models. I want to thank my lab. Everybody in my lab worked really hard on different projects, especially big shout out to Neha and Skanda, who are the pivotal personal force to push the project forward. I also want to thank all my collaborators at Drexel, around the world in UMass and Texas to help me. Without that collaboration, my research won't get get this far. I really want to thank NIH and SPF Foundation to give me the resources to study SPG4 and HSP. And also a big shout out to the foundations led by the parents of the childhood onset SPG4, the Cure SPG4, the Lydia and Blair Foundation, and Maury Coterie Foundation. They connected me with the different scientists around the world to build up collaborations, to talk to companies, and then I can't thank them enough for me to connect with the different scientists. The last but not the least, I really want to thank Spastic Paraplegia Foundation to give scientists a platform to interact and meet patients. I think this is so essential. I have been to this meeting only from last year and this year. I can't tell you enough how important that is, this interaction and meeting is, to meet with the patients and patient families. I had a lot of good conversations with the patients and patient families, and really kind of inspired me, on different levels about my research, about the questions I should answer, I should try to answer, and transforming sort of what can feel like dry and routine bench work as a scientist in the lab to really motivating and impactful endeavors to try to make things better for you guys. I mean, absolutely, that's really, really essential to kind of to push the therapies, to push the disease mechanism studies towards what we really want to be. I can't thank enough about this platform provided by the scientists and interacted with the patients. So I stop here and take questions, if any.
Okay, Oscar, I got one right over here.
Excellent presentation. Thank you so much. Every year you present very good data, and
Thank you.
It's exponentially growing, so thank you. Maybe I didn't understand, so if you could clarify for me. So you said HDAC6 could be a therapeutic target. So do you want to reduce it? Do you want to upregulate it? Do you want to improve interactions? What kind of a target do you envision, and based on which result?
Yeah, based on, first of all, we find HDAC6 was hyperactive. And we did Western blot, look at the protein levels. The increase of the protein levels is moderate. So we don't have a lot of proteins in there to make it hyperactive. What I think, we don't have a good antibodies to look at phosphorylation and modification on this protein, but we have a really reliable kit to look at the functions of the protein, which means the activity of the protein. We see the activity is highly increased in our SPG4 animals. Now we also replicated that in our brain organoids derived from SPG4 mutations. So I think as a target, we need to turn it down. We need to turn it down. And also, as HDAC6 has been shown in other diseases, for example, the Parkinson's, as well as the Alzheimer's, the hyperactivity of the HDAC6 is also in those diseases. Okay. So there are companies actually are developing HDAC6 inhibitor drugs to treat cancer, to treat Alzheimer's, and also I know there are a couple of companies, they also are trying to use HDAC6 or other HDACs to treat ALS. So we can probably connect with them to see whether we can collaborate with one of them.
Yeah, but then be careful, because the activity may be a compensatory mechanism. Because it's active doesn't mean it's toxic. So it could be that the cell or the system activates it to overcome some of the challenges that it has. So
that's
everything that goes up in a high expression doesn't mean it's bad. So it may be it may be that you actually may want to increase it even further to help. So, just because it's high may not mean that's bad.
Yeah, that's not the case in our experiments. When we treat with the inhibitor, which tubistatin we got from the company, and then we see the degenerative markers turn down. So,
Good.
Yeah, so that's we mark with the SMI32. So I think that's kind of promising. And also I didn't present it here. We have animal models collaborate with Dr. Bass and using his animal models, and we see when we inject IP with HDAC6 inhibitor, tubistatin, the same drug we use in organoids, the behavior of the animals were rescued, as what Emanuela was showing with all the parameters with the catwalk. So we think that's actually a positive side.
Excellent.
Thank you.
So for the organoids, I'm going to ask a time question. Let's say if someone walks in the clinic and then you said these are derived from fibroblasts. Some of
Yes.
them are fibroblasts. Okay.
Yeah.
So let's say you take a little bit of the fibroblast from a patient or from a person. So you develop organoids. What is the time frame? How long does it take going into clinic, giving a fibroblast?
So from the fibroblasts to the organoids, so we turn fibroblasts into iPS, the stem cells, within a month. And then we have to validate everything, like no mutations introduced, the chromosomes are normal, and things, just quality control, take another month and a half, probably. And then after that, then we can expand it, and then we can put into our liquid nitrogen tank for unlimited use in the future. But now we can start to differentiate into organoids. So the organoid differentiation takes, the younger ones takes a month to get to the younger, really, really young organoids, very teeny tiny little things growing in the bioreactor. But we managed to grow them out for 12 months. So we can stop in whatever time you want to do longitudinal studies with a different age of the organoids. What I presented here mostly are 6 months, 3 months to 6 months old organoids. So that's the timeline we have for the human stem cell models.
And then from one fibroblast, it's just one organoid, or you have hundreds of
Oh,
organoids?
hundreds.
Hundreds of organoids.
So each generation with our bioreactor unofficially counted probably 60 to 90 little organoids growing on. You can see
And
the
all those
spinning.
organoids are similar to each other?
Similar. They're very similar in the size. We have this really wonderful bioreactor in the lab that can consistently generate the size comparable organoids that kind of mitigate a little bit about variations between batch to batch.
Excellent. So then going from fibroblast to having your first young organoid is about 4 months.
Yes.
And then you can grow them for an extension of a year.
Yeah.
That's actually very nice because you can use all these individual organoids for drug discovery purposes, maybe.
Yeah, that's the goal, is to generate as many as we can and to do a lot of screening on those.
Ding, ding, ding.
Well, there you go, guys. You get to see researcher to researcher interaction right there, live in person in this room. That was pretty cool. We're going to do another question back here.
Thank you so much for that presentation. I thought it was actually surprisingly easy to follow. So really, really appreciated all of that. And also very exciting. I mean, it sounds like, significant progress in, SPG4. So I mean, I have a couple of questions, like you mentioned with this HD6. Well, first question is there anything else that you see in the cells, or have you studied, anything else in the cells that's showing up that could also be, causing the degeneration? And it sounds like this theory with HDAC6 did pan out, and you're seeing the results with this. You said it's a statin treatment?
Tubistatin.
Okay, tubistatin. So then, okay, so the first question is there anything else? The second question is, what are the next steps? Because it seems promising, and you found a drug that addresses it.
So yeah, love these two questions. The first one is we actually are doing very, very unbiased high throughput analysis on the organoids on our isogenic lines. combining the proteomics and single-cell RNA sequencing, we have several druggable targets we recently discovered. We're collaborating. I'm not an expert on some of the targets. I can't elaborate too much here because, with the time constraint. I collaborate with some people I can find around the country, like we're expert on these targets. We're actually doing more analysis. I submit some grants just to try to see whether these are actually promising targets for future testing. That's one thing. The second question I think you have is what's the moving forward? What's the strategy and things like that? I actually talked to two companies, and one of them I think the Leland Blair Foundation, the funder Chris here connected me with the ALS the drug and the developed ALS drugs. They actually have HDAC drug. They actually doing that with the patients with the ALS. So we recently talked to them, and then there's some kind of nuanced things we need to take care of. But I think to talk to the pharmaceutical companies or biotech companies to repurposing whatever they are using for testing or for already approved is a way to go. And then that could be really fast and testing with the really, really early phase 1 studies on these compounds, whatever new compounds they have to inhibit HDAC6. The other thing is I also talked to big farmers like the Merck and Pfizer just to see whether in their portfolio do they have drug of interest. Although these big farmers, they're concentrated on big disease like Alzheimer's and Parkinson's, but they might have something they can also repurpose in terms of treating HSP, SPG4. So I'm kind of anybody who has connections, feel free to contact me. I would like to talk to people who have, the information like that, just to moving forward. We want to put into clinical trials as soon as possible.
Great, everybody. Thank you very much. Dr. Chung, thank you, sir.